So this is what happens during the charging reaction. After that, after the charging reaction, after the charging reaction, we have the binding of messenger RNA to ribosome and chain initiation. The binding of messenger RNA to the ribosome and chain initiation. So remember the start codon, which signals initiation of translation, is AUG, which also encodes the amino acid methionine. So usually the ribosomes are going to use the first AUG it encounters in a messenger RNA strand to signal the start of translation. And then we have proteins that are called initiation factors that are also involved in initiation. So what happens during this process of initiation and binding of messenger RNA to ribosome? So you have the messenger RNA, which is going to bind to the small subunit. So in prokaryotes, it's a third S subunit. So the messenger RNA is going to bind to the small subunit or the third S subunit, which will lead to a formation of a complex called the third S initiation complex. And then what will happen is that that third S initiation complex binding with the 50S initiation complex, or rather 50S ribosomal subunit, will lead to the formation of the 70S initiation complex. So like we said, we have the small subunit binding to messenger RNA at the AUG start codon. And then we have initiator transfer RNA that binds to the start of the codon, and one of the initiation factors has to be released. And then when that large subunit binds to a small subunit and the remaining initiation factors are released, we have the 70S initiation complex, and henceforth the initiation complex is complete. So the process of initiation simply begins when a section of ribosomal RNA in a small subunit binds to a complementary sequence on messenger RNA. The messenger RNA region is what is called the ribosome binding site, or the shine diagonal sequence. So a small subunit is positioned correctly on the messenger RNA due to a conserved sequence in the 5' end of the messenger RNA, which is known as the ribosome binding sequence, which is complementary to the 3' end of the small subunit of ribosomal RNA. So the role of initiation factors is simply to help in preparing the ribosome for translation, which includes the binding the first amino acid tRNA to the ribosome. And also the initiation factors prevent the small and large subunits of the ribosome from coming together until the initiator tRNA is in place at the AUG start codon. And they also help bind the messenger RNA to the small subunit of the ribosome. So initiation factors are quite important. Initiation, as we have said, is complete when the large subunit joins the complex, forming the 70S initiation complex. So once the complex of the messenger RNA and the initiator tRNA and the small ribosomal subunit is formed, the large subunit is added and translation can begin. So the formation of the complete ribosome the initiator tRNA is bound to the P site, the peptidyl site. We have the initiator tRNA. And then the A site, amino acid site, will be empty. So we have AUG on messenger RNA, and we have the anticodon, which is UAG. So during initiation, we have the initiator tRNA that is bound to the P site. And so the A site will be empty. So in summary, when we are saying, when we are talking about translation initiation, it is a 3-step process. So we have messenger RNA binding to the small ribosomal subunit. And then we have the initiator amino acid tRNA, which bears the F-methionine or formylmethionine in prokaryotes, binding to the start codon. And then we have the binding of the large subunit of the ribosome, which completes the initiation complex. When we look at initiation in eukaryotes, it's quite complex than in prokaryotes. However, it still involves recognition of a start codon. So in eukaryotes, the start codon is methionine, or in prokaryotes, the start codon is N-formylmethionine. And then we still have the assembly of the ribosome. So the ribosomes has to come together. And then we have the assistance from initiation factors as well. And then we also have the positioning of the methionine carrying transfer RNA in the P site. However, in eukaryotes, we have methionine as the initiator tRNA, which is unmodified. Because when you look at prokaryotes, we have the modified methionine, which is F-methionine. And then we also have the cape and poly-A tail, which are also important in assembling the ribosome on the messenger RNA in eukaryotes. And also eukaryotes lack the RBS, or the ribosome binding site, as we mentioned earlier. Then the small subunit of the ribosome to the messenger RNA initially by binding to the 5' cap of the messenger RNA. So the 5' cap, as we looked at last time, is quite important in the recognition of the binding of the small subunit of the ribosome. So after initiation of chain elongation. So during chain elongation, we have the polypeptide chain, which is covalently bonded to the transfer RNA, carrying the amino acid most recently added to the chain. So we have this transfer RNA in the P site. The transfer RNA in the P site. And then we have the amino acid transfer RNA binding to the codon in the A site in the presence of energy, GTP or ATP. This will lead to an amino acid transfer RNA that will bind to the A site by complement base pairing between the anticodon on the transfer RNA and the messenger RNA codon. And then we're going to go to the next process, which is peptide bond formation. So that growing polypeptide chain will detach from the transfer RNA molecule that is in the P site, and that will become attached by a peptide bond to the amino acid that is linked to the transfer RNA at the A site, or the amino acid site. And then we have the next step, which is called the translocation step. So during the translocation step, the ribosomes move one codon towards the 3' end of the messenger RNA, resulting in the growing polypeptide chain being transferred to the peptideo site. And then we have that unchanged tRNA. The unchanged transfer RNA that is in the exit site or the E site will then exit the ribosome. And then that will make the ribosomes to be ready to accept another amino acid transfer RNA. So the process of translocation does several things. One, it moves the uncharged transfer RNA into the E site. So it moves the uncharged RNA into the E site. Secondly, it moves the transfer RNA that is containing the growing polypeptide chain into the P site there. And then it opens the A site and exposes a new messenger RNA codon. So it does several things. And then the other things that translocation does is that empty tRNA that binds itself in the E site is then ejected from the ribosome into the cytosol or the cytoplasm. And then we have the chain termination. So the process of translation will continue as a cycle. It's cyclic. So it continues until the ribosome reaches a stop codon, any of the 3 stop codons. So what will happen is that the A site, the amino acid site, will bind to a protein release factor. That release factor will then hydrolyze the bond between the polypeptide chain and the transfer RNA, which will cause the release of the polypeptide chain from the transfer RNA molecule into the P site. So the polypeptide chain is released. And then we have the remaining part of the translation complex. So that we need to disassociate. So of importance during the chain termination phase are the release factors and the stop codons. So when you look at a termination or translation, in summary, we are saying when a termination codon is reached, and when that stop codon occupies the amino acid site, release factors will cause the newly synthesized polypeptide to be released from the ribosome. So the ribosomal subunits will then dissociate from the messenger RNA. That is, the ribosome will be detached from the messenger RNA and split into its subunits. So you have the large subunit and the smaller subunits being dissociated from the ribosome. So the release factors fit tightly into the amino acid site. They have a size and shape that is similar to tRNA containing or coming into the ribosome. But however, these release factors do not carry any amino acid. So when they occupy the A site, the protein's active site, that protein's active site, they will cause the hydrolysis of the polypeptide bond. So this reaction will lead to the freeing of the polypeptide. And then after translation, we have post-translational modifications. So for the proteins to be fully functional, fully formed and fully functional, they need to be modified. So they have to go through extensive series of processing steps, which we call post-translational modification. And of importance is the folding of the proteins into a three-dimensional structure. So post-translational modifications of importance are the folding. So a protein's function really depends on its shape, and the shape depends on how it folds itself. So for the protein to be functional, fully functional, it needs to fold into a three-dimensional structure. So it does this by the help of what are called the molecular chaperones, which assist in the folding of the newly synthesized polypeptide chain. Then the other modifications are chemical modifications. So chemical modifications simply entails the addition of chemical groups or functional groups. It also entails proteolytic cleavage of the polypeptide chain. So under chemical modifications, we can have the addition of carbohydrate functional groups. We can have the addition of phosphate groups, which will lead to phosphorylation. We also have intramolecular bonding, such as hydrogen bonds, that can happen during chemical modification. We also have processes such as the addition of the acetyl groups, leading to a process that we call acetylation, and other functional groups. So after these have been added to the proteins or to the polypeptide chain, the protein then can become functional and fully formed.